Scientists today announced a landmark plan to recreate entire human cells from scratch within the next 10 years. The enormously complex project involves synthesizing all 6 billion letters of the entire human DNA code, otherwise known as the genome. If the project goes ahead, it could have far-reaching implications for the study of diseases such as cancer and even growing replacement organs, say researchers. But the work could also heighten existing public concerns over a fast track to designer babies. Scientists are hoping that the completed DNA, once it's made, will be implanted into a living cell and, it is hoped, start to divide. At this point scientists will have created, for the first time, a whole human cell of their own design. Named the Human Genome Project, right, it could enable researchers to make synthetic human genes and chromosomes for study. This could include chromosome 21, an extra copy of which is responsible for Down's syndrome. But implications could extend far beyond, to growing organs for transplant patients, engineering immunity to lethal viruses, such as Ebola or Zika, and even developing cancer-resistant cells in the lab. Writing in the journal Science Today, researchers explain that plans to create a synthetic genome would be the next step on from sequencing of the human genome, which was completed in 2004. Led by Professor Jeff Boak of New York University, the authors write that HGP write would be a natural progression and would switch research from a reading to writing mode. The group's paper follows in the wake of closed-door meetings on genome editing held at Harvard Medical School last month, with scientists, lawyers, entrepreneurs and government officials, but from which journalists were excluded. One of the main goals outlined by the group is to slash the cost of engineering man-made genomes within cells in the lab, reducing the cost 1,000-fold over the coming decade and making the approach more affordable. The team say it would be an expansion of techniques already used in the lab to edit human and animal cells, but scaled up considerably from making small amounts of synthetic DNA. The 25 experts who outlined their aims said they initially need $70 million funding to launch the project. They said the final cost was difficult to estimate but would likely be a little more than $2 billion, which was the cost of the Human Genome Project. Boak claims the technology that needed to be developed to synthesize the human genome could also be used to construct animal and plant genomes, such as those of pigs and mice. Possible applications include being able to grow, in pigs, human organs for transplantation. By being able to write long sections of DNA code, it should be possible to genetically alter organs so that they are not rejected by the human body when transplanted. Scientists say that the project will require consideration of ethical, legal and social implications from the start. Scientists have already created a synthetic life form. Called Mycoplasma laboratorium, it had only around 580,000 DNA base pairs compared to around 3 billion in a human. So to create an entirely synthetic human cell is vastly more difficult. The 4 building blocks of DNA, adenine, guanine, cytosine and thymine, are difficult to work with as they become increasingly brittle as the strands of DNA get longer, making them more difficult to manipulate. To achieve the aim of synthesizing the entire human genome will require a huge leap forward in techniques used to replicate DNA, assemble it into long strands, and to implant it in a cell. But just as computers became hugely cheaper and more powerful in a short amount of time, scientists believe that the technology will rapidly advance. Under guidelines outlined in today's paper, the initial stages would see a number of pilot projects carried out to explore techniques and make small chunks of the genome, around 1% of the total. However, the authors explain, it will require public involvement and consideration of ethical, legal, and social implications from the start. One of the main arguments against research into editing of the human genome has been that any changes could be passed on to future generations through the germline, in sperm and egg cells. At the end of 2015, this led scientists to call for a moratorium on the tinkering with the DNA of any cell which can be inherited by the next generation, which had been demonstrated in non-viable human embryos by a Chinese team earlier in the year. But the group has outlined security measures for ultra-safe cells with edited genomes, making it impossible for them to pass on changes down the germline. Experts reacting to the news have said that the proposal is just the start of an open and transparent debate on the topic of creating a man-made human genome. Work is already underway to create a man-made yeast genome, with 2 groups in the UK involved in creating synthetic chromosomes as part of the project. The latest proposals would be an extension of the research to humans, and an increase in the scale. Researchers not involved with the group have said making man-made DNA will enable scientists to uncover the role of elements in the human genome without a clear role, and why there is so much variation from person to person. Dr. Nathan Richardson, head of molecular and cellular medicine at the Medical Research Council, told Mail Online, "To our knowledge there have been no serious discussions within the UK about contributing to such an initiative. A major international initiative such as this would need substantial funding commitments from multiple countries. Whether it is necessary to establish an international consortium of the likes of the Human Genome Initiative is a matter for debate." Dr. Richardson also highlighted the need for responsible research, in line with existing legislation, which will need to evolve as the technology advances. Hugh Whitall, director of the Nuffield Council on Bioethics, told Mail Online, "This is a highly speculative article about future possibilities in genomic research, and we will keep an eye on whether and how it develops. As the authors note, it will be important for the potential ethical considerations to be addressed from the earliest stages." He added, "This should be an inclusive, public and transparent debate that considers the public interest in research policies and priorities, potential applications, as well as possible risks and regulatory responses." The Nuffield Council on Bioethics is carrying out a consultation on human genome editing and expects to publish its findings later this year.